Hello everyone and welcome. My name is Rafał Wójcicki. I'm a biologist and genetist, and for more than a decade I have been involved in the design and bioinformatic analysis of genomic experiments in various biological contexts, from plants to animals to clinical research. The topic of my talk is the practical planning of genomic experiments. How to move from a biological question to a well-chosen technology and ultimately obtain data that truly answer the stated hypothesis. I would like this presentation to be practical and supportive, regardless of whether you work with animal models, cell models, or in clinical studies. Let's begin with a brief overview of what we will discuss today. First, we will talk about how our understanding of genetic variability has changed from the days when we focused almost exclusively on SNPs to the point where we realized that true genetic complexity lies elsewhere. In the second part, I will try to show that the most important stage of an experiment is asking the right question. This is what defines the technology we should use. The third part will be a review of current sequencing technologies, what we have at our disposal today, what they are suitable for, and what they are definitely not. Then we will move on to the principles of experimental design, what affects the reliability of results, what is often overlooked, and what is really not worth cutting corners on. And finally, a summary, questions, and hopefully an engaging discussion. Over the last 15 years, we have undergone a major shift in thinking in genomics. In the so-called early NGS era, we focused on SNPs and small indels, mainly because that was all we could reliably detect with short reads from platforms such as Illumina. These data were very valuable. They enabled the development of association studies, identification of candidate genes, and so on. But they had one serious drawback. They covered only the most unique parts of the genome, that is, mainly exons. Today, in the long read era, we know that this was a very limited perspective. The true variability, the kind that plays a key role in evolution, gene regulation, and complex diseases, often lies in places we used to ignore, in repetitive sequences, introns, and regulatory elements. Moreover, genomes are not simply different versions of the same book. Within the same species, they can have different structures, different gene copy numbers, completely different chromosomal rearrangements. That is exactly what we are beginning to see today, thanks to long reads. Conversations about technology often start at the wrong end. Do we have the budget for PacBio? Or will Illumina be enough? And I always ask, what exactly do we want to study? Because that question, and only that question, should determine the choice of technology. We need to answer a few fundamental points. First, are we interested in information at the DNA, RNA, or epigenome level? These are three completely different layers of biology and require entirely different methods. Second, what scale of variability do we expect? Are we looking for single mutations or large genome rearrangements? Third, do we want to see an average from the entire cell populations? Or do we need single cell resolution? The answers to these questions are crucial because they determine not only the choice of technology, but also the way we prepare samples, the statistical design, and ultimately whether the results will make sense. This is a scenario we know very well, and one that works perfectly. If we want to compare a group of patients with controls and find new SNP variants or small insertions deletions, Illumina is still a gold standard. That's because it provides very high accuracy at the single base level, which is essential when analyzing point mutations. Another big advantage is cost. The unit cost of sequencing at the base has dropped dramatically and today allows the analysis of large cohorts, which 10 years ago was beyond the reach of most projects. But we have to remember the limitations. This data will tell us little about repetitive regions, large genome rearrangements, or haplotypes. So if our question concerns structure rather than just individual changes, Illumina may not be enough. Sometimes our research question goes beyond point mutations and concerns the entire genome structure. For example, when we want to assemble the genome of a new species or detect large chromosomal rearrangements invisible to short reads. In such cases, long reads become the technology of choice. PacBio HiFi is currently the gold standard when we need accurate genome assembly and high-precision structural variant detection. It provides long reads at very high accuracy. Oxford Nanopore, on the other hand, offers extremely long reads, even over 1 million bases, which allows the assembly of the most repetitive and problematic genome regions, such as centromeres. It also has the advantage of enabling field sequencing on devices such as MinION. It is also worth mentioning Hi-C technology, which adds the third dimension, showing how fragments of chromatin interact spatially. This not only helps in assembling the genome to the chromosome level, but also allows us to study genome organization in the cell nucleus, which is important in stress responses and diseases. When our goal is to compare gene expression between groups, for example, to find out which genes change expression levels after stimulation or treatment, we usually turn to RNA-seq on the Illumina platform. This approach is very well established. It allows us to obtain high-quality quantitative data at a relatively low cost, enabling the analysis of many samples and the inclusion of biological replicates. A major advantage of this approach is the availability of excellent statistical tools such as Dissect2, Limma, or EdgeR, which provide a solid foundation for detecting expression differences. However, we have to remember that when using short reads, we obtain the sum of expression for all variants of a given gene. We cannot see how individual isoforms, different mRNA versions resulting from alternative splicing, change. In some cases, this may be a limitation. We often say this gene has increased expression, but that is an oversimplification. One gene can have several, sometimes a dozen isoforms, different mRNA variants, which can perform completely different functions. Sometimes one isoform acts as an activator and another does the opposite, as an inhibitor. Therefore, if we care about precisely determining which isoform is changing, we must use long-read technologies. PacBio IsoSeq provides highly accurate full-length transcript reads, allowing unambiguous assignment of a read to a specific isoform. The downside is that it requires converting RNA to cDNA, so we lose information on RNA epigenetic modifications. If we want to preserve native RNA, we should consider Oxford Nanopore direct RNA sequencing method. This is the only available technology that can detect modifications such as M6A in RNA. We just have to remember that raw accuracy is lower. But we can compensate for this with deeper sequencing or specific error correction. The genome is just the beginning. Regulation is controlled by the epigenome. It means changes not written in the DNA sequence, but influencing gene expression. And today, we have tools to analyze many such layers simultaneously. For DNA, the oldest method is bisulfite sequencing, which identifies 5'-MC methylation, but degradation of the material and lack of information on other modifications are serious limitations. New approaches, such as native DNA sequencing on PacBio or Nanopore, allow detection of various types of modifications without the need of amplification. When it comes to RNA, again Nanopore stands out with the ability to detect modifications at the level of individual RNA molecules. For chromatin, We have techniques such as ATAC-seq, which tells us where chromatin is open, and ChIP-seq, used for mapping protein binding sites and histone modifications. And finally, single-cell genomics. This is no longer just a trend, but increasingly a necessity. We can analyze not only gene expression, but also full-length isoforms or chromatin accessibility in a single cell. However, it's worth remembering that this approach is still costly and requires specialized platforms. In this part, we move on to practice. Even if we choose the best technology without good quality input material, nothing will come of it. For long-read sequencing, it's crucial that DNA is of very high quality, high molecular weight, intact. This means no vortexing, no pipetting back and forth 10 times, no repeated freezing and thawing. For RNA, integrity is the most important. The RIN parameter should be above 8. This gives us confidence that RNA has not degraded. Here too, fast handling, the use of RNase inhibitors, and sticking to the standard protocol are important. It's worth remembering that not only the material itself, but also the way samples are collected and stored can decide the experiment's success. Especially for RNA, a delay of just a few minutes can drastically change the expression profile. No matter how exciting our scientific question is, in the end we always come down to two basic issues: budget and statistical power. Every technology has its cost. And it's not just the sequencing cost, but also library preparation, data processing, and analysis. That's why it's worth realistically matching the platform to the study's aim and available funds. On the other hand, data alone is not enough if we don't have the right number of biological replicates. This is not optional. It is an absolute necessity if we want to draw reliable conclusions. The minimum is 3 replicates per group, but if we expect subtle effects or have high sample variability, we need more. And finally, statistical power. If we run a low-powered experiment, even if the effect truly exists, we will not be able to detect it. What's more, we may encounter random results that appear significant, but are actually false positives. That's why power needs to be planned in advance, not checked afterwards. There are 3 things without which no genomic study makes sense, and no technology will make up for it. First, a clearly formulated research question. This question drives the whole experiment. It tells us what we should measure, with what technology, and in what biological system. Without the question, we just have a random collection of data. Second, proper controls. And not just the classic ones, negatives or positives, but also biological and technical. We need controls to distinguish a biological effect from random variability, technical errors, contamination, or protocol differences. Third, biological replicates. I mean truly independent samples. Not 3 libraries from one sample, but 3 separate organisms, cultures, or clinical samples. Only then can we assess variability and calculate meaningful statistics. One of the most common reasons why genomic data cannot be interpreted is so-called batch effects, technical differences between samples processed under different conditions. This can be different days, different people, different reagent lots, or different machines. The problem is that these differences can look like a biological effect and mislead us. That's why planning is so important. Randomization, randomly assigning samples to batches, group balancing so that each batch contains an equal number of samples from each experimental group, protocol standardization, the same equipment, the same reagents, the same settings. And one more thing, documentation. Record everything. Who, when, with what, for how long, on which machines. because if a strange effect appears without this information, we will be not able to interpret or correct it. Power analysis is one of the most important, yet most underappreciated tools in experiment planning. It asks a simple question. With this number of samples and this variability, do we have a chance to detect the effect we are interested in? To perform it, we need a few things: expected effect size, for example, a twofold change in gene expression, an estimate of variability, for example, from previous studies, and a chosen significance level, and desired power, usually 80%. The result of this analysis is a specific number, how many biological replicates we need, and this helps us make real decisions, whether to reduce the number of comparison groups or increase the budget. For RNA-seq, there are many tools available, including my own online power calculator. I strongly recommend using it. Before spending tens of thousands, it's worth making sure the experiment makes sense. The last key technical parameter is sequencing depth. How many reads we generate for a given sample. And again, it all depends on the goal. If we are only interested in highly expressed genes, moderate depth is enough. But if we want to detect rare transcripts, new isoforms, splicing events, we need much higher coverage. In metagenomics, the situation is even more challenging, because we have many organisms and a very complex mixture. Here, depth increases dramatically, especially if you want to reconstruct complete microbial genomes. Also remember that for isoforms, the signal is distributed. If one gene has 5 to 10 transcript variants, each gets only a fraction of the total reads. Therefore, when planning transcript-level studies, depth must be greater than in standard RNA-seq. Here we have a summary of the whole process. The path from a biological question to results we can interpret and use. We start with a scientific hypothesis. This could be a question about gene expression, variant presence, response to environmental conditions. But it must be a specific, measurable question. Then, we define the study's objective. Are we looking for changes in DNA, RNA, epigenome? Are we interested in point changes or structural ones? And are we analyzing the average response or single-cell heterogeneity? On this basis, we choose the technology: Illumina, PacBio, ONT, maybe Hi-C or ATAC-seq. Each has its applications, strengths and weaknesses. There is no single best one, only the one best suited to the question. Then comes experimental design. Here, all the issues we discussed earlier come into play: material quality, number of replicates, batch effect control, power assessment, realistic budget. And only then, when we have a well thought out and executed experiment, do we move on to bioinformatic analysis and biological interpretation. And it's worth remembering, success doesn't start in the sequencer. It starts in the planning step. Technology is just a tool, a powerful one, but one that requires conscious use. Thank you for your attention. I hope I have shown that good planning of genomic experiments is not just about choosing a platform. It's a process that starts with a question and goes through many conceptual and technical decisions before we even make the first read. If you have questions about the technologies or the practical aspects of planning, I invite you to contact me. I'm also happy to talk about how genomics can support your specific research projects. On the slide, I'll also provide a link to an up-to-date list of sequencing costs, which may be useful when planning budgets. The data are current as of June 2025 and based on US sources. Thank you once again, and I look forward to your questions.